In this video, we'll be talking about the humoral branch of the adaptive immune response. Now, when we're talking about humoral immunity, we're talking about what B cells do. And B cells, when they get activated, they become these little factory cells called plasma cells. And what plasma cells do is produce antibodies. That Y-shaped structure, that's what an antibody looks like. This diagram walks you through what happens when a B cell actually encounters antigen and becomes activated. So the first time a B cell encounters antigen, we have the dark blue B cell here binding its antigen. The B cell proliferates. It divides and divides and divides and divides to form a clone. A clone is just a set of genetically identical cells. This process is called clonal selection because you are selecting for the B cells that have actually encountered antigen. Let's talk a little bit more about this. So to illustrate clonal selection, say you've got 3 B cells, each one with a different receptor. We have our red receptor, we have our blue receptor, and we have our black receptor. Now, a blue antigen comes in, and by a blue antigen I just mean an antigen that the blue B cell receptor can interact with. Now, the B cell with the black B cell receptor isn't seeing its antigen, so it's not going to proliferate. The B cell with the red B cell receptor also isn't seeing its antigen. It's not going to proliferate. But the B cell with the blue B cell receptor does see its antigen, and it makes a huge number of copies of itself, all with that blue receptor. Now the nifty thing is that as it divides, it can actually get a little bit better at recognizing the antigen. So there's a selection process in which the B cells that are the absolute best at binding that antigen, they get a little bit more stimulation, and you have even more of those. This is the process of clonal selection. Basically means that by the time you are reaching adulthood, you've encountered tons of different pathogens, tons of different bacteria and viruses. Your B cell population is a bunch of different pools of these clones, these genetically identical, more or less genetically identical B cells that have all encountered pathogen. Okay, so you have clonal selection taking place, and you have this big population of B cells that are all capable of seeing your antigen. Now, the majority of these B cells are going to become what are called plasma cells, and plasma cells are the effector B cells. And by calling them the effector B cells, that just means that they're the actual active B cells. And plasma cells are the antibody-making factories. They're kicking out loads of antibodies. Now, the majority of these activated B cells become plasma cells, but a small proportion don't. There's a few of these activated B cells that don't become plasma cells. Instead, they become memory B cells. And these memory B cells, although they aren't actively producing antibody, they're very long-lived. See, plasma cells are short-lived. They die pretty quickly after the infection is cleared. But memory B cells, they stick around, and they can stick around for years, decades even. But they have this really neat capability in that they are ready to go. As soon as you encounter that same antigen again, the memory B cells recognize it, they get activated, and they proliferate to make new plasma cells. Again, the majority, on that secondary response, the majority of the daughter cells become plasma cells, but a few become memory cells. So if you keep seeing the same infection over and over, you're eventually going to have more and more memory cells hanging out, ready to go, so that you can attack that infection more strongly and more quickly the next time you see it. It's these memory cells, as you might imagine, that are responsible for humoral memory. So there's this basic pattern here that you see with B cells. First seeing antigen, then proliferating, and then becoming effector and memory cells. This general pattern, you see it with both B cells and T cells. Those three steps, seeing antigen, proliferating, and forming effector and memory cells, holds true for T cells as well as for B cells. It's just that with T cells, the effector cells do different things. They don't produce antibodies. There's actually a very important step in B cell activation that was left out of the previous diagram, and that is T cell help. B cells actually need two signals in order to become fully activated. Signal 1 is antigen recognition, right? So first of all, you need to have the B cell receptor binding its antigen. But signal 2 is T cell licensing. This is where the T cell help comes in. It's kind of like the B cell is getting permission from the T cells to actually respond and become fully active, become plasma cells, and become memory cells. The reason why you have this, or the reason why it makes sense to have this, is because T cells go through a much more extensive selection process to make sure that they tolerate self-antigens. The selection process for T cells happens in the thymus. And the B cells, they undergo some selection, but it's really not as extensive and not as rigorous as the T cells. So this T cell licensing is basically a check. It's just making absolutely sure that the B cell isn't attacking something from your own body. If the B cell is recognizing a self-antigen, because that B cell receptor has escape tolerance, it's not going to get any help from T cells because there won't be any T cells that can recognize that self-antigen. So this T cell licensing step is critical for self-tolerance. Okay, now let's talk a little bit about the antibodies. You should recognize the term immunoglobulin because immunoglobulin is just another term for antibodies, the globular proteins of the immune system. Antibodies have this Y shape. They've got two long, heavy chains and two short, smaller, light chains. And the end of each kind of arm of the Y is the point at which the antibody binds antigen. Okay, so there's an antigen binding site on this arm and an antigen binding site on this arm. And these are referred to as the variable regions because they are basically different for every single antibody, or for every single B cell. Every single B cell produces antibodies with a different variable region, at least before you have clonal selection. At the base of the Y, you have the constant region. This constant region is what determines the function of the antibody. Different classes have different constant regions. So the constant region determines what class an antibody is, which basically determines what that antibody is good at. This is also the region that interacts with phagocytes or complement. Interacts with white blood cells. Remember the classical pathway of complement activation? It's that constant region that's recognized by complement proteins. And if you remember the Fc receptors that phagocytes have that enable antibodies to do opsonization, that C stands for constant, because it's this constant region or constant fragment that the receptors on phagocytes bind to. Now let's talk a little bit about the different antibody classes. Let's talk a little bit about what different constant regions allow antibodies to do. The first two antibody classes I want to talk about are IgM and IgD. And I'm talking about these two first because these are the first two antibody classes that are produced. So both IgM and IgD can act as B-cell receptors. So I've said the B-cell receptor is basically just a membrane-bound antibody, and to be more specific, it's a membrane-bound IgM or a membrane-bound IgD. Okay, and that's all IgD really does, is serving as a B-cell receptor. IgD is not secreted as an antibody, but IgM can be secreted as an antibody, and when it is secreted, it's secreted as a pentamer. Now the prefix penta means 5. So the IgM pentamer is 1, 2, 3, 4, 5 IgM antibodies all joined together by a covalent bond. Because it's got 5 antibodies, it has 10 antigen binding sites. So having those 10 antigen binding sites all stuck together means that one of the things IgM is really good at is agglutination. Agglutination is just the clumping of antigen together. It makes it easier to phagocytose. It also makes it harder for the bacteria or virus to actually interact with your cells if it's all clumped together in a big lump. So IgM is great at agglutination. It's also good at activating complement, stimulating that classical pathway. And because IgM is the first immunoglobulin class released, it's a good indicator of active infection. If you're wondering whether someone actually has whether the infection is still present in someone's body, if it's a relatively new infection or if it's still there, you can look at the levels of IgM in someone's blood. High levels of IgM indicates that the infection is actually still there and there are still new B cells getting activated. The other 3 antibody classes are IgA, IgG, and IgE. IgA is secreted as a dimer. So you have, dimer means 2, and you have 2 antibodies joined together, again, by that covalent linkage. The main function of IgA is to protect body surfaces. IgA is actually found in body secretion. So it's found in sweat, it's found in saliva, and it's found in breast milk. So this is the antibody that's actually passed from mother to breastfed child as a form of naturally occurring passive immunity. IgG and IgE are both found as monomers, so just one antibody. IgG is the most abundant antibody. The vast majority of the antibody circulating in your blood is IgG, so 75 to 85% of the antibody in your blood. IgG has a number of different functions. IgG crosses the placenta, so it helps protect the fetus in utero. IgG is also good at activating complement in the classical pathway. And IgG is really good at neutralization. So neutralization is basically just binding up viruses or bacterial toxins so that they can't harm your cells. The reason why IgG is good at neutralization is because IgG is usually produced later in the immune response. So remember, I mentioned briefly that B cells, even after they're activated, even after they've seen antigen, there's some changes that are made to the antibodies, and they undergo a competition process so that only the antibodies that are the absolute best at binding antigen are produced in large volume. And since IgG is produced a little bit later in the immune response, it's gone through that selection process. So IgG antibodies tend to be much better at binding antigen, and that's what makes them so good at neutralization. IgE is an antibody that you probably have a love-hate relationship with if you have allergies. Actually, it's probably mostly just hates. You have these cells in the tissues of your body, underneath the mucosal surfaces, called mast cells. And those mast cells are filled with inflammatory granules. And those mast cells are just sitting there waiting for stimulation to release all of those inflammatory granules, including histamine. Right? Now, mast cells carry an Fc receptor that specifically binds IgE antibodies. So, if you make IgE, it sits in the Fc receptor of the mast cells, waiting for the antigen that IgE antibody recognizes to come along. And when that antigen comes along, that stimulates the mast cell, and you get release of the inflammatory mediators, release of histamine, and you get all of the lovely symptoms that we're so familiar with. Now, I mentioned that the first antibody class produced is IgM and that shifts to IgG over the course of an infection. Let me show you how that relates to memory. That's what this graph is showing you. The memory response to an antigen can be followed by looking at antibodies and looking at the specific type of antibody. So in a primary response, on your first exposure to antigen, you make a small amount of IgM first, and then over time, you make greater amounts of IgG. So you switch to IgG. But on the secondary response to an antigen, now most of the memory cells that are left are going to produce IgG. So in the secondary response, you have a really strong, fast IgG increase in your blood. And this is IgG specific to that antigen. Okay? But you're still getting new B cells activated. So if you're exposed to antigen, you'll have the memory cells producing IgG, but you'll also have new B cells getting activated and producing IgM. So you'll have a low level of IgM as well. And if you were looking at a patient and you wanted to know, well, is this a first response, or is this something they've seen before, and is it a response to something that's already gone, or is it still there? You can look at the antibody titer, which is just a measurement of the total number of antibodies in the blood, but you can also do a titer specific for IgM and one specific for IgG to discriminate between a primary and a secondary response, and to see whether there's an active infection. Now, we've talked a little bit about some of the various things that different antibody classes can do to antigen, to take care of antigen and remove it from the body. I just want to lay it all out for you. These 5 things are what can happen to an antigen after the antibody binds it. So these are the results of antigen-antibody interaction. Now, opsonization, we already talked a bit about when we talked about the innate immune system, because this is a point at which you have the adaptive immune response, you have the antibodies from the adaptive immune system strengthening the innate immune response, enhancing phagocytosis, right? Complement, we also talked about when we talked about the innate immune response, because again, another point where antibodies are helping out the innate immune responses. This is the classical pathway of complement activation. Agglutination, neutralization, and antibody-dependent cell-mediated cytotoxicity, ADCC for short. These are new, so let's talk about these in a little more detail. So agglutination is just clumping together antigen, like the clumping that occurs with red blood cells when you're running a blood typing assay. So remember I said IgM is very good at agglutination because there's 10 antigen binding sites on a single IgM pentamer. This is showing you an example of that. See, there are 3 different bacteria that are all kind of stuck together, bound together by IgM. It's like IgM is the glue that's holding them together. So what this IgM has done is it's taken 3 bacteria and kind of clumped them together into a single unit. So that clumping together makes phagocytosis a little bit easier because now there's just one particle that needs to be taken up. It can also keep the bacteria from attaching to your cells and colonizing your cells and setting up an infection. And because some of the attachment points that the bacteria would need may be kind of hidden away in the middle of this clump. Agglutination is also really useful for diagnostics. So here we're looking at an E. coli, a type of bacterium. And this, by taking antibodies that you know are specific for this O157 antigen, it's a particular protein on E. coli, you can mix it with the E. coli, and see those dark little clumps show that this is in fact an E. coli O157. And if you take antibody against the H7 antigen, mix it with the E. coli, you see the clumps that indicate that the E. coli has the H7 antigen. So you can demonstrate that you have this particular strain of E. coli, which is the particular strain of E. coli that can cause kind of a nasty, bloody, fatal diarrhea. Neutralization is exactly what it says. It's neutralizing neutralizing a virus or neutralizing a toxic molecule produced by bacteria. Basically, if you are looking at a toxin or virus, when you combine that toxin or virus with your cell, your cells die. But it requires an interaction between the toxin and your cell, or an interaction between the virus and your cell, in order for your cell to be affected. If you have antibodies against that toxin or against that virus, the antibodies basically bind up the toxin. They coat it so that it can't interact with your cell. And now your cell is protected. Okay. And as I've said, IgG is particularly good at neutralization. It tends to bind more strongly, and it tends to be more specific. The 5th outcome of antibody binding antigen is ADCC, antibody-dependent cell-mediated cytotoxicity. This happens when you have a pathogen that's just too big to phagocytose, or too big to be taken care of in any other way. Now, when you have an antigen like this, you can still make antibodies against it. The antibodies bind the surface of the parasite, coat the surface of the parasite. And as they coat the surface of the parasite, their FC region sticks up like a little flag. And various cells, eosinophils, macrophages, and NK cells can come in, recognize that FC region. And that acts like a trigger for them to release all of their worst damaging enzymes. So you get inflammatory cytokines being released, you get lytic enzymes being released, perforin enzymes, basically just trying to punch holes in the cells of this enormous parasite and kill it, right? So you get an inflammatory reaction and you get lysis. It's like this is too big for these cells to phagocytose, so they kind of vomit out their digestive enzymes onto the surface of the parasite and attack it from the outside in. Okay, so these are the five things that antibodies can do to antigen. Just to summarize, we've got agglutination, opsonization, neutralization, complement activation, and ADCC. These questions give you some idea of what you should be able to address after studying this video. You should be able to talk about how B cells are activated. So remember the two-signal theory. You need both antigen recognition and T cell help. You should be able to talk about both plasma cells and memory B cells, where they come from and what they do. You should be able to talk about clonal selection, and remember, T cells do clonal selection as well. You should also talk about how antibody classes change during a memory response, and talk about what the different antibody classes do. So in general, the ways in which antibodies protect against infection, and then what each antibody class is more specialized for.